I spoke before about the issue of the COVID-19 epidemic, but in particular in relation to the so-called second wave. And I've been talking about this as an epidemic of tests rather than an epidemic necessarily of illness or an epidemic of death. And so that's what I want to talk a little bit about this evening. The testing for COVID-19 is done using a thing called a PCR test. So much of what I'm going to talk about is about PCR testing. And I am quite certain that this presentation is going to bring up as many questions for you as it will be necessarily providing answers. Because this is really a very interesting and tricky area, but one on which is extremely important. It's important in particular in relation to this issue of false positives. And I spoke before about how these false positives are very closely related to the number of tests carried out. In other words, when you carry out vast numbers of tests, then you're going to get considerably large numbers of false positives. So these PCR tests they're a kind of a genetic test, and every organism has got a kind of a genetic makeup, which is a kind of has got a kind of a series of genes, and these are made up of a chain of particular little molecules. And so the PCR test is something which is designed to pick up on a certain sequence. So we pick a little sequence that we think represents the thing we're looking for, and that's what we search for, and that's what the PCR test does. It's very hard to pick out individual little molecules, little pieces of DNA or RNA. It's very hard to pick them out. So what happens is that the test, what it does is it multiplies these out. So in order to make them detectable, the test, it runs through a number of cycles, and each cycle will multiply the amount of bits and pieces in the test by about 1,000. So if you increase the cycles, you do what you increase the CT value. So for example, a CT value of 10 means that you've gone through 10 cycles, so you've multiplied what's in here by 1,000 times, and you've done it 10 times. So you can go from having a small number to having maybe billions of little pieces, and that makes them easy to identify and makes them easy to find. So if your test is positive after a very small number of cycles, it means that there must have been a lot of viruses there to begin with. So in other words, the fewer the number of cycles, the greater the number of viruses and the more infectious somebody is. So for example, if you had a PCR test and you were positive and there was 10 cycles, it means you're very infectious. If there was 30 cycles, then it's a lot more questionable as to whether you're infectious at all. I'll talk a bit more about that. So, as I say, these cycles multiply the number of particles so that they're more detectable. They reckon that anything over 30 to 35 cycles starts to make the test pretty well meaningless, because, you know, at that stage, you're talking about producing trillions of particles where there were virtually none there to begin with. They reckon that anything over 30 to 35 cycles makes a test invalid. How many cycles do they take routinely when we have ourselves tested at home? Nobody knows. They don't tell us. The WHO, their standard, or what they say should be carried out by laboratories, is 45 cycles. I'll talk about this in a few minutes, but it has been shown very clearly by many scientific studies that anything approximating 45 cycles has no validity whatsoever as far as the test goes. So if you get a positive test and don't have a CT a number with it, then you know it really lacks meaning, and you don't really know what way you are. So these are the daily figures from a certain date, and you can see 430 new cases. What were the CT numbers? That's extremely relevant, and there's no indication. Um, the government, or certainly the Irish government, seems to equate a case with a positive PCR test. Um, uh, a case is not necessarily a sick person, and 430 cases is not 430 sick people. What is it? What does it represent? Actually, we don't know, right? It could either represent nothing or what the hell, right? So this just is a diagram showing how the PCR test works. It changes the RNA into DNA, and then it multiplies that out, doubling or up to 1,000 times between each test, and until you get, as I say, this huge number of after cycles. So there's been quite a lot of stuff in the literature showing that anything over 35 cycles should be disregarded. And that the CT value is what tells us whether somebody can spread the disease or not. Anything 13 to 17 would suggest that somebody is a spreader. Anything greater than or equal than 34 is of no value. A lot of the studies of patients with positive tests, for example, this particular study identified 800 positive tests based on 40 cycles, and that would 70% of them would no longer be positive if you limited the cycles to 30. In this other study, 90% of the patients who were tested positive would not be positive if the threshold of the tests had been limited to 30 cycles. So as I say, there's this strong inverse correlation between the infectiveness and the cycled threshold value, a value which we don't know, but we are led to believe is in the realm of the 40s. The original paper, which described the test, which is used, the PCR test, is called the Corman-Drosden paper. And again, there was no standard operating procedure outlined in this paper, which again is clear that anything above 30 is likely to be false positive. As I say, this is more really about this Corman-Drosden WHO protocol, which unfortunately talks about 45 cycles being carried out for a PCR test. And I'm sorry, but that's really not something that can be given any validity. So just to summarize, anything greater than 35 is of no validity. Anything between 30 and 35 is suspect. Anything over 40 is garbage, and anything over 45 has to be considered beyond garbage. So as I say, we've got problems with false positives relating to sample size, as I mentioned earlier. And you also have a problem with false positives in relation to the number of CT cycles that are carried out. What's the threshold that's standard for these laboratories? Is it less than 30 or is it greater than 30? That is very, very important and significant. So let's just dig a little bit deeper and talk about how this how does the test work and what's it derived from? I mentioned to you about these little bits of DNA, these little sequences that are identified. Every living organism has got these as DNA. The human has it contained inside their chromosomes, and a human being has 23 pairs of these chromosomes. And these contain 6.4 billion pieces of information. That's, in other words, it's a sequence of 6.4 billion pieces of information. So visualized, these little dots are these pieces of information, these nucleotides as they call them, and there are 4 different ones and they're arranged in a different sequence. So in the same way as in a computer everything is ones and zeros, in living organisms there are actually 4 of these: A, C, T, and G. So you've got this whole kind of garbage mix of A, C, T's, and G's that go for, as I said, so many billion of these. And this is what they work out when they work out what was called the human genome. They basically work out what this whole sequence is. So that's an example of a small little piece. So if we identify a piece that we say, look, there's a unique little piece here, if we're going to look for that. And if we find that, then we found a particular type of organism. So that's really what the test is based on. So the virus, it contains these chains of RNA. And what we do is we isolate a little bit of that and we say that's what we're going to look for. And so how do they find the sequence that they actually look for? And as I say, how do they test for it? So when the virus was, when the disease was found, they isolated material from the patient, and they identified what they felt was a new virus, and they found that this virus had an 80% similar sequence to the SARS-CoV virus, which had been around a couple of years previously. And they did this using what was called de novo assembly, right? And this is a way of, what to say, constructing genomes from a large number of fragments where there's no knowledge of the correct sequence or the order of those fragments. So this was how they constructed the genome for the SARS-CoV-2 virus, and they call it MN908947.1. And this, as I say, had an 80% match to the SARS-CoV virus, the previously known one. As yet, no one has provided a single purified sample of the SARS-CoV-2 virus. And what we actually have is a genome which has been made from fragments. So it's actually a theoretical model rather than a real one. So this is an example of a section of the genome of the SARS-CoV-2 virus. And what they have done is they've taken 3 parts of it, and they call them 3 genes, 3 parts of it. And each part they've taken a little sequence from, and these are what they search for. So these are the sequences on the right-hand side, the orange little sequence of letters. And as I say, there are 3 of these. They're called the S gene, E gene, and the RNA polymerase gene. And so when you have a PCR test and you're positive, that's because they've found these markers. They've found these, and they say, right, you've got it, right? We've found this. The tests do not test for all 3. They would generally test for 1, and some at times they test for more than 1. When you have a sequence like this, there's a search engine online that has got the genetic code for virtually every organism that's ever been sequenced. And you can enter in this sequence, and you can find out, you put the sequence in here, and it'll say, oh yes, SARS-CoV-2, right? Because that's what the sequence is seen in the organism of SARS-CoV-2. However, if you do enter it into the search engine, it will reveal 99 human chromosomes in which this sequence 100% matches. In this E gene, there are 90 human chromosomes which have 100% sequence identity match. There are also 100 microbes that have got a 100% sequence match in both the E gene and the RDPRP gene. Why is this a problem? Well, it's a big problem actually, because it basically means that these sequences that have been selected, and dare I say it, arbitrarily selected from these gene sites, do not have specificity for the SARS-CoV-2 virus. That's the S gene, similar situation. So the issues we have then really are, first of all, that the SARS-CoV-2 genome was assembled from fragments. We actually don't know what it is. We actually don't know it, really. The other thing is that the fragments that are currently being used to identify it, they are not unique to SARS-CoV-2. This is a problem because we are equating positive PCR tests with new cases of infection or of disease. So not only are we uncertain about whether the identification is correct, but we also are worried about the correct attribution of a positive test. So if you have COVID-19, I assume at least that you would be able to identify these 3 genes in it. Let's work backwards, right? Well, this Moroccan study, tested COVID-19 patients and found that only 9% were positive for the 3 genes, 18% for 2, 73% for just 1. So what's this telling us, right? It's really telling us that the tests that we're carrying out for this disease may have very little relevance. The test itself, how did it come into being? Well, we know that COVID-19 raised its head in early January. I think it was January the 9th or the 12th. The test was first described in a paper on the 23rd of January 2020. The article itself was submitted to the magazine 2 days prior, 21st. And on the 23rd of January, it had recognized scientific status. With the publication of the article, the test became the scientific standard for COVID-19 detection. This came from a paper which had no peer review, and such a thing takes weeks or takes months. And this particular paper, which I'll talk a little bit about in a few minutes, had some very, very serious flaws. And this paper is the basis on which the entire edifice of COVID-19 testing stands. This paper, which appeared in October 2020, reviewed the Corman-Drosten paper, which is the one which I just described, the one that introduced the idea of the PCR test. This paper was produced by a very large number of very high-standing scientists. And they talk about 10 fatal problems with the Corman-Drosten paper, which they deal with in this particular review. And I just very brief, you can get this paper online. It's just sitting there for you to see. And so what it really talks about are the fact that, first of all, that uh, the sequence, the genetic sequence for the coronavirus is basically a theoretical sequence, which has been provided without control materials of infective or non-infective SARS-CoV-2, and no validation has been performed based on isolated viruses or a full length of RNA, because they haven't done it. Similarly, they're talking about the time scale between the submission and acceptance of the publication and the lack of peer review. Similarly, the scientific and methodological blemishes which they talk about, which I'm not going to go into seriously, but there are 10 of them outlined in the paper. So just to finish up then, what are the issues with the PCR test? Well, I suppose the biggest issue and the one that could just finish the whole discussion once and for all is, does it actually have any meaning at all? We know that there are false positives due to the large sample size. We also know that false positives are going to come because of dead viral fragments, but even bacterial fragments and even human DNA fragments are going to give positives with this test. The lack of any CT values, which has to do with the number of test cycles, the lack of any information in relation to this, again, in my view, completely invalidates the PCR test as something that has any real relevance in relation to knowing the level of disease that's present in society, or what stage we're at. So as I said, this presentation raises as many questions as it does provide answers, but I do think that the questions that it raises should cause you all very, very serious concern, and it should make us wonder the information, who has known this information, how long have they known it, and why have they continued using what they call the gold standard PCR test to enforce society's response to what is clearly a disease with a considerable effects and mortality in society. So thank you.